Good morning, everyone, and thank you for attending today's session. My name is Vicki Lightbound. I am the Director for the Water Innovation Program here at Alberta Innovates. While we're waiting for everybody to get allowed into the system, I'm going to go over a few housekeeping items. At the bottom of your screen are some engagement tools that you can use. All the engagement tools are resizable and movable, so feel free to move them around and get the most of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit them through the Q&A tool. We'll try to answer as many as we can at the end of the webcast. The group chat box is a useful tool to connect with other participants. I would encourage you to use the tool to discuss the session topic, but please don't let it distract you from the presentation. For the best viewing experience, we recommend closing any programs or browser sessions running in the background that could cause issues. You can find additional answers to some common technical issues located in the help tool at the bottom of your screen. With that, I'd like to welcome everyone to the Alberta Innovates Water Innovation Connect series, community-based monitoring and public outreach session. People and communities connect on a personal level with the water systems around them. Recreational waters are a great example of how people and water interact. However, there are often barriers to people accessing those waters. A combination of real-time information and education programs can help break down some of these barriers. Today, you'll hear from Patrick Hannington with the University of Alberta and Catherine Wagner with Insight Education. Their bios are available in your engagement tools. For those of you who are not familiar, Alberta Innovates is a provincial corporation created to support research and innovation activities. We provide funding programs, advice, connections, technical expertise, and applied research services. Our scope encompasses the whole innovation journey, from generation of ideas through applied testing and validation to commercialization and end use. This Water Innovation Connect series will share ideas and outcomes from projects funded through Alberta Innovate's Water Innovation Program. The Water Innovation Program is designed to help achieve the goals of Alberta's Water for Life strategy and Alberta's Water Research and Innovation strategy. The knowledge and technologies developed in this program will help to create a clean tech industry in water treatment, support improvement in water use conservation, efficiency, and productivity, and provide safe, secure, and reliable water resources for a growing population while maintaining the health of aquatic ecosystems. I hope this series will provide you with some valuable information and spark discussion. Now let's get started. I'll pass it over to Patrick, who will be speaking on his project, integrating molecular detection testing into an Alberta-wide community-based water monitoring and research program.
Great. Thanks, Vicki. And hi, everyone. I'll do my best to look up at the camera every once in a while. My slides are down a little lower, and I'll be doing a bit of up and downing with my eyes just to make sure I can see what's going on. And I'll try to make sure that I'm conscious of highlighting for you what I'm trying to talk about on each slide, because I don't think you can see my pointer. So like Vicki said, I'll be talking to you today about some of our work related to amplifying DNA in communities and schools. That's sort of the abbreviated title that I decided to put on this slide. And I just wanted to emphasize that while we have a lot of partners that are identified on this title slide, the most important one here has been Alberta Innovates. They've supported this work for a number of years and have been really fantastic in helping us get a lot of our community-based programming out in Alberta, and that's nucleated a whole bunch of partnerships throughout North America that have allowed us to undertake some really exciting projects related to monitoring for various biological organisms in water. And I'm going to highlight two really sort of our kind of flagship stories that I think really emphasize the value of community-based monitoring programs for water monitoring. So just to clear up everything at the beginning, I'll talk about community-based monitoring, or CBM, on a number of my slides, and really that's sort of a synonym for citizen science, which is another common term used for this type of work. While they're used interchangeably, I think that the term community-based monitoring is gaining more favor now within the community rather than citizen science for a couple different reasons, but keep in mind that they're basically the same thing when we're talking about it here for this talk. And I wanted to quickly just highlight that this type of research, community-based monitoring or citizen science, is something that we have been using for a number of years to help advance our understanding of a variety of different organisms and ecosystems and biological systems. And so some great examples are things like the Christmas bird count, or iNaturalist, which is an app that you can get for your phone where you can take pictures of different things that you don't know what they are, and somebody will help identify them for you. We have a variety of different marine water monitoring programs. The Eyes of the Reef in Hawaii is an example, but we have similar programs that exist around the world to monitor reef ecosystem health or marine ecosystem health in general. Here we have things like the Swim Guide, which allows people to understand recreational water quality parameters throughout Canada, and that's a great example of another one that has apps that you can use to help upload your own data or report your own system to them, and then they can be a source of information back. Here in Alberta, maybe our best known and one of our longest lasting community science programs is run through the Alberta Lake Management Society, which has a long history through the Lake Watch program of doing citizen science and community-based monitoring. And even within academic circles, we are starting to see the more sort of research-based programs be integrated into community science to help expand the capacity of those programs to understand more about our world. And so this is just one example of a relatively recent publication that talks about eDNA and citizen science and integrating eDNA-based monitoring into the detection of invasive species, which is something that we're going to talk about today. So we have all these different examples, and I want to just emphasize that there are a couple different models that we can follow when we implement a community-based monitoring program. The first model, which I think most community-based monitoring programs really fall into, is this situation where a researcher is designing a project and interpreting results, and they rely on community partners to expand the scope of their program. And so here you might have community partners collecting samples or doing something for the researchers. But the data typically will reside with the researcher, and they'll analyze it and write up a paper or something like that. And this is fine. I'm not trying to suggest that Model A isn't a good model, but it's one in which the community-based partners are typically working as an extension of the research team. Model B only differs really from Model A in the sense that that there's more of a sharing of the data. And so in this model, we see researchers designing the project and interpreting the results, but we also see community partners collecting samples and sharing the data, so that the community partners have a little bit more skin in the game, per se, in this situation where there's a little bit more data sharing on the side of the researchers. And that can be important because it can help guide future research questions and tailor those questions to maybe be more relevant to the community partners. Model C, which is, I think, in theory, the one that many community-based research projects try to strive for, but often struggle to achieve, is one in which the research and the community partners are both designing the research project and both involved in interpreting the results. And in many cases, there's a partnership for sample collection, which still often relies on the community partners to do the sample collection. But the data is shared between the researchers and the partners, and there's often a refinement of the question based on that data. And so we see these various models, and there's sort of a gray area in between each one. But within the world of community-based monitoring, or participatory research, as it's sometimes called, we see these sort of stratifications that allow us to get an idea of the role of the community partner compared to the person who's organizing the project. And no matter what model you're working on, there's really an agreement that there's about 5 main principles that need to be followed and adhered to in order to make the most out of these kinds of partnerships. We need to have really strong engagement of participants. We want to have strong and frequent communication. We want to have some sort of contribution to science, and the goals need to be clear. And in order for all these things to be true, we also then need to be generating reliable data. And sometimes this is really the fifth purple circle here, is where we sometimes see a falling apart of community-based programs, depending on the type of data that's being collected and the level of interpretation that's required by the community partners to collect that data, we can sometimes see pushback from the scientific community during the peer review process, for example, on the validity or the reliability of community gathered data. And so it's really critical that when these programs are being developed, that the data reliability issue be addressed. And so we see community-based monitoring programs being part of water monitoring programs quite frequently. And these have been things that have happened for many years. And it's very likely that some of you have been part of something like this in the past, where you may take a water grab sample for a cyanobacterial bloom monitoring program, or you might take a zooplankton sample, or you might do a hydrolab assessment using a water probe of some sort. Or you might be using something like a Secchi disc to measure water turbidity or sedimentation stuff. What we tend to not see as frequently as part of community-based monitoring programs are things on the right, which include some of the more advanced technologies for measuring water hazards from a rec water perspective or a drinking or stormwater perspective, the molecular biology tools that are important for getting very high precision, high accuracy, and high sensitivity for various organisms, and some of the microscopy-based techniques that rely on a trained eye to be able to identify, for example, a veliger larva from an invasive mussel. And all of these types of techniques, in one way or another, are prohibited from being part of CBM programs. One of the main reasons is because of the financial cost of integrating those into those programs, which can be quite substantial. For example, the qPCR machine in the upper left corner of this right-hand box can cost upwards of $30,000 to $40,000. And so that's not something that you will see easily implemented as part of a community-based monitoring program. No matter what, though, when we're looking at the types of things we might be interested in monitoring water for, especially in this context, recreational water, we can see that a number of different targets emerge, and they relate to things like health, economics, and environment. And so we can stratify those targets and how we might approach detecting them with all those different tools I showed you on the previous page in a variety of different ways. So, for example, there are health targets like the fecal bacteria, fecal coliforms, enterococcus, or specific microbial source markers like HF1A3, which is a human fecal marker. They relate mostly to the health side of things. And we have those that stratify based on health and the environment, like saprozoic pathogens, environmental pathogens, or things like Legionella that causes Legionnaire's disease and in-premise plumbing. We have some that are economic and environmental, like invasive species or species at risk or fish populations. In Alberta, things like whirling disease fall into this category. And then we have some that touch on all three elements in some way, cyanobacterial blooms as an example. And how you might monitor for each of these different things as part of a normal monitoring program or even a community-based monitoring program could really be quite different. You might look at a cyanobacterial bloom through a grab sample and just do a quick cell count analysis, but you could also do a toxin analysis that requires a mass spec. Or you could be using molecular or culture-based techniques for enteric bacteria. So there's a huge range of potential ways to detect these different targets. And that also is prohibitive from integrating monitoring for some of these different organisms into a community-based program. But what's nice, and what we have been advocating for as part of our research, is that all of these organisms on this slide are biological, and they're all alive. And because of that, they all have DNA. And because they all have DNA, we can unify around a single technique, which is called quantitative polymerase chain reaction, or qPCR, which is actually a pretty old technique. It's been around for a number of years, but it's just now being integrated more commonly into official monitoring programs for various different types of targets. And as we've seen eDNA or environmental DNA emerge as a really appealing tool for monitoring for things like invasive species or species at risk, we've started to see DNA-based monitoring becoming a much more commonly used tool for environmental monitoring purposes in general. So because all of these different organisms have DNA, we can use qPCR to detect all of them. We just need to know enough about each one of them so that we can design new tests or leverage tests that have been already developed to integrate them into a monitoring program. So, well, I said that these images on the left are part of our traditional community-based monitoring gear for water-based monitoring programs, the real crux of what we're proposing here is something slightly different, which is that now with the advancements in qPCR technology, particularly with respect to bringing the price of qPCR down significantly, we can start envisioning ways to integrate qPCR into water monitoring programs in a much more democratic way that doesn't rely on them being part of core laboratories, but that can be things that we place in people's garages at lakes or in offices somewhere, that allows us to decentralize the technique and be much more regional in terms of how we approach monitoring for various targets. And so on the right here, I show you the open qPCR platform, which is made by a company called Chai Biotechnologies. And they've partnered with us on a couple different projects. And they produce this machine, which is pretty sturdy. It's pretty heavy, but it's much cheaper. It runs around $5,000, and it allows us to run a smaller number of samples, but is very easy to use, very user-friendly, and was designed really for integrating qPCR into schools, which is something we're going to talk about later on. But because of this technique and the reduction in cost of getting qPCR out there, we've been able to envision ways that we can use qPCR for a much more decentralized monitoring approach. And so for today's talk, I wanted to highlight a couple different stories for you, and I'm just going to walk you through them pretty quickly. I'm going to try to emphasize the takeaway message with respect to how we couldn't have done these studies without the community partnerships that we've ended up making. That's really what my take home for you is, but I'm going to also try to tell you something cool about the organisms we're talking about as well. So I'm going to tell you a story about research, which is about tracking swimmers itch causing parasite populations in lakes. I'm going to tell you a story about monitoring, which is a really exciting development that's happened really just recently in 2020 about monitoring enteric bacteria at recreational beaches in Alberta. And then I'm going to quickly touch on using qPCR in an educational context for water and molecular biology teaching and also research. And that's where I'll pass it off to Catherine at the end to emphasize that in a much more defined way. So the research talk I want to talk to you about is about implementing qPCR for swimmer's itch parasites, which if you've ever watched a presentation I've given before for an Alberta Innovates talk, this is one of my passions is these flatworm parasites that penetrate through the skin of a variety of different animals and cycle through snails. And in Alberta and North America in general, they cause swimmer's itch. And then in other countries, in more tropical regions of the world, some of these species can actually cause human disease. But this has been a really exciting progressive research project that has really allowed us to undertake some really, I would just call them insane studies on this parasite group. And where we've learned the most actually has been in Michigan. And so this is a project that's really been a big spin-off of our Alberta Innovate supported work, which has allowed us to really engage with a bunch of lakes in northern Michigan to learn about swimmer's itch at a really high level. So I'm going to introduce you to the situation there, which is very similar to the situation here in Alberta, but I hopefully can sort of walk you through the progress of this project I'm going to explain to you in a few slides. So I started working in Michigan with some of my students in 2016, and when we started working there, the current story was this, where we had this waterfowl, the common merganser. It hosted a parasite that was reliant on this snail, Stagnicola amarginata, and the parasite that would be released from that snail was called Trichobilharzia stagnicolae, which everyone was pretty sure was the main species causing swimmer's itch in Michigan. And to give you some context, swimmer's itch was discovered in Michigan, and it is very, very bad there. They probably have the worst swimmer's itch in the world, and so it was kind of a perfect storm for us to be working on swimmer's itch there because there was a bunch of people who had a number of different properties at these very beautiful lakes in northern Michigan, and they were upset because they couldn't have their kids or their grandkids swim in the water because they would get really bad swimmer's itch. And so when we started working in Michigan, we were very surprised by this sort of one-dimensional story related to swimmer's itch because in Alberta, we have many, many species that can cause swimmer's itch here. And so we thought initially that this was a really interesting opportunity to understand possible ways to control swimmer's itch with much less complexity than what we have here in Alberta. But really quickly, some of our first experiments that we did there were just to confirm that this single parasite was really the main causative agent of swimmer's itch in Michigan, and we realized that it wasn't really the case. And so very quickly we discovered that there was 2 other snails that were cycling parasites that were using the common merganser as a host. And this one snail here, Limnaea stagnalis, was also cycling this Trichobilharzia stagnicolae parasite. And then the phycid snail, which is a different genus of snail, was cycling a different parasite, Trichobilharzia phyceli. And if we included other bird hosts in this equation, we found that the mallard was cycling a whole bunch of other species of parasites that could cause swimmer's itch. And if we included the Canada goose, we found that they also had a bunch of other parasites that could cause swimmer's itch. And we actually, as part of this survey, the parasite at the very, very top, the Trichobilharzia nov species, that parasite was actually a previously undescribed species. So we actually ended up identifying new species of parasites as part of this survey. And this one actually, as you'll see, ends up being a very important swimmer's itch causing parasite in Michigan. But if we take a step back, while we were identifying all these different parasite species in Michigan, the rest of the people in Michigan were basically trying to figure out ways to get rid of the common merganser. And so what ended up happening there is that they developed a program that relocated the common merganser from all of these northern Michigan inland lakes to the Great Lakes. And so they would trap baby mergansers as they came off the nest. They would trap the brood, including the mother, and relocate all those birds to Lake Michigan or Lake Huron. And so it was a really ridiculous ecological study that was being done in the context of swimmer's itch control. And so thousands of birds were relocated over a number of years. And because of that, we were trying to advocate for the fact that this might not be a really effective control measure because we knew about all these other species. And so we decided that we would design a study that attempted to determine whether or not the common merganser relocation program was really effective. And to do that, we targeted development of qPCR assays at the parasites that you can see the stars next to, because we knew these were very prevalent in the waters in Michigan through a big survey that we did. And it was pretty easy to distinguish them from all the other different species. And so our goal here was to essentially do a large-scale ecological study where we chose lakes that had either chosen to relocate common mergansers, or lakes that had chosen to not relocate common mergansers, or lakes that didn't even have common mergansers as resident birds. And we wanted to understand whether or not the relocation efforts actually impacted the population of that one parasite, Trichobilharzia stagnicolae. And so we did this as in a study that we called the 3 Lakes study. And it was a much more comprehensive study than what I'm going to show you, but the 3 lakes as a part of this study were kind of the most core lakes in the study. So we had 3 lakes that I just want to introduce you to here because they probably aren't familiar to you. We had South Lake Leelanau, which had no resident mergansers that were over the summertime, and it had no control program. And we had Lake Walloon, or Walloon Lake, which had merganser broods, but they also had no control program. And then we had Glen Lake, which has merganser broods and had been relocating mergansers for 3 years. And so this was really our experimental lake. They, in theory, had no resident mergansers for 3 years. And so what we were interested in is really understanding whether or not the migratory birds that come through Glen Lake and all of these lakes in the spring and fall might be contributing enough parasite material to the water that they could, in theory, be continuing the life cycle of Trichobilharzia stagnicolae. And so what we ended up doing was within this 3 lake study, like I said, we had many other lakes that were participating at some dimension in this study, but the 3 lakes were the main ones. And I wanted to highlight just the metrics of this study because this is an example of a really incredible citizen science project. We had a total of 16 lakes that were in our study, and we had chosen 10 sites per lake to evaluate for swimmer's itch parasites. And in at least those 3 lakes that I just showed you, and at a couple other lakes as well, those sites were sampled every week between 8 a.m. and 10 a.m. By a community partner. And so we had a single sample collected from every one of those sites with time being essentially removed from the equation, because we know time can impact when we see these parasites emerging out of the snails. We had 2 coordinators here at the University of Alberta, myself and a student, Sydney Rudko. And we had 6 program managers on the ground in Michigan trying to coordinate over 25 community partners who were collecting these samples. And so between May and November, we collected over 500 water samples. We ran over 1,000 tests for just general swimmer's itch. And then we used the results of those tests to target down to our species-specific qPCR tests. That meant we ran about 5,000 individual qPCR tests for different species of swimmer's itch causing parasite. And what that really translated to was a huge amount of effort, over 800 hours of community partner volunteer time, 100 hours of qPCR runtime, and 1,000 hours of processing and sample collection time, over 640 hours of summer student time in Michigan working on this project, and over 200 hours of qPCR validation in our U of A lab. So a really monumental effort, and one that I think I'll emphasize here and I'll emphasize again, couldn't have been accomplished without all the community partnerships doing this kind of a study, especially the fact that we had samples collected at all these different locations between 8 a.m. and 10 a.m. That would just be actually geographically impossible because it takes more than an hour to drive between these different sites. So I'm going to take some time and walk you through what we learned from this study. Before I do anything, I just want to orient you to what these heat maps actually are showing you. So if you look at the upper left heat map from Glen Lake in the blue spots, the blue dots, on the bottom axis of the heat maps is the date of the sample collection. And then on the y-axis of the heat map is the sample location site, so the site that was sampled. And so each of these different dots represents a single location at a single time. And then for each of the heat maps, there's a gradient color scheme with the darker the color is, meaning essentially that there's just more parasite in that sample of each of the different species. And the species are identified by color. So blue is the T. stagnicolae parasite, which is the one that we know goes through the mergansers. Then there's the T. novel species, which is the one that goes through the Helosoma snails and the Canada goose. This is the new species that we identified. And then there's Trichobilharzia ficeli, which goes through the phycid snails and often is going through mallards. And then you can see we've broken down these heat maps based on lake. And so this just gives you a sense for the scale of the data just from these 3 lakes. It's a huge number of data points that have been collected. And at each of those different time points, we have a huge amount of metadata that include things like water temperature and wind direction and things like that we can now apply to this data set. So there's a few take-home messages that I want you to get from this. Clearly, you could spend a lot of time just looking at this and trying to tease out your own ideas. But the main thing that we concluded is actually just shown to you in the upper right or upper left, heat map from Glen Lake. This is the lake that did the relocation effort for 3 years. And that parasite in the blue, T. stagnicolae, is the one that they were trying to remove from the lake, the one that they thought was causing all of their swimmer's itch. So clearly that didn't work. T. stagnicolae is the dominant parasite at Glen Lake, even after 3 years of merganser relocation. And so this suggested to us that the migratory birds were contributing enough parasite material to that lake that it was persisting, that parasite there, which was it was a relief in a way because Glen Lake was spending a lot of money relocating those birds. And so now they don't have to anymore because they know it was essentially ineffective at removing that parasite. We also discovered, if you look at the center at Walloon Lake in the novel species, which is the red heat map, we discovered that there are a number of lakes where this new species of parasite, which we had never known about before, was actually the main parasite causing swimmer's itch. And that became problematic because that's going through Canada geese, which are very, very prevalent in northern Michigan. And so that became a problem for control efforts in general because clearly you couldn't just focus on one bird like the common merganser and deal with all of these parasites. And then the final take-home message that I think we can emphasize is that there are certain times and certain locations where we found there was very infrequent detection of any of the species of swimmer's itch causing parasite, which you can see, for example, on most of the heat maps on Glen Lake in the early and the late time points, so on the very far left and the very far right of the heat maps. And then if you look at individual sites and just work your way across on the heat map, you can also see that there are certain locations that very infrequently have swimmer's itch. And so we've actually replicated this study in 2020. This is all 2019 data. And so we have another year of data that we'll be using to understand whether or not those, for example, site-specific trends are true. And maybe those sites are ones where we would recommend people actually go swimming rather than some of the ones where there's a whole bunch of parasites. And I just want to emphasize that this study wouldn't have been possible without the community partnerships. So we went to Michigan and we trained a bunch of people who then trained a whole bunch more people to do qPCR. This is one of the labs that we set up, and you can see my back there talking to people as other people are learning qPCR. And we'd set up labs. In particular, this is with our partner group there, Freshwater Solutions, who has essentially a fully functioning qPCR lab now. And they get to go out on these nice lakes and boats and collect water samples and collect snails and understand snail density patterns and collect. This is the collecting of the water sample. And this is what happened every morning, every Tuesday for basically the entire summer from May to November last year and this year. So this is, I think, one of our sort of flagship stories about community monitoring. The other one that I wanted to introduce you to is an Alberta story that happened this year in 2020, where we were working with various partners to supplement the beach monitoring program here in Alberta to collect samples to understand about wreck water safety with respect to enteric and cyanobacteria at Alberta beaches. And so in Alberta, we actually have developed a really comprehensive suite of qPCR tests for a variety of different targets relevant to the province. We have tests now that we use pretty routinely for various invasive species, the parasite that causes whirling disease. We've developed a test for proliferative kidney disease, which is another fish parasite that is just south of our border right now, so something that we maybe like to catch before it gets here. Chinese mystery snails, which is the image that I'm showing you in the background here, is a giant snail that we now have in McGregor Reservoir here in Alberta. And then various invasive mussels or fish. We also have done a lot of eDNA monitoring now for various, again, species at risk, invasive species, or even doing paleo eDNA monitoring where we look at core samples from lakes and go back in time looking to see when we can detect the DNA from different target organisms. The big difference here is that for these invasive species tests, we're actually looking for the actual organism in many cases. In the eDNA test, we're looking for DNA that's been shed into the environment by these organisms. And then we have the biological hazards that we like to look for, and these include things that are included in a variety of different official rec monitoring programs, things like enteric bacteria like Enterococcus, which is the newly sort of minted rec water guideline target for beaches in Alberta. And that can then lead to microbial source tracking testing for various fecal contaminants that come from specific organisms like humans, ruminants, gulls, or geese. And then we also have cyanobacteria testing that we do for total cyanobacteria and the microcystin toxin gene, the MCYE gene. And this story that I'm going to tell you really focuses on, I'm just going to talk just about the Enterococcus test, which is the enteric bacteria test that we've been running, although the program that we've been running is actually slightly larger than just the Enterococcus. And this program, again, I'm going to kind of try to summarize it with this sort of summary slide, but it has relied really heavily on a partnership with the Alberta Lake Management Society to help coordinate all the sample collections from a whole bunch of different locations in Alberta. And so they've been a really critical partner in all this. And I can't emphasize enough how much they've done this summer to help with this project. And they also are the location where we've set up a satellite qPCR lab, which ran all of these samples as part of this study. So we can see here that we ran 68, we sampled 68 different lakes as part of the enteric bacteria program. We took beach and pelagic samples from those lakes as well as others, looking for enteric bacteria at the beach samples and pelagic with cyanobacteria. And all those samples were coordinated mostly through the Alberta Lake Management Society, or ALMS, but we did a bit of coordinating ourselves through partners that we have through our labs at the U of A. In the end, for just the enteric, it meant over 240 samples were collected for the beach program, over 2,000 qPCR reactions were run, and we looked for 4 different targets, 2 cyanobacterial targets and 2 enteric bacteria targets. What was really unique about this study, though, and why I think it was a really important one to talk about today, is that, as I mentioned, one of the big criticisms of community science has always been the validity and reliability of the tests. And qPCR allows us to try to address some of those issues because we can implement a number of different types of controls and things like that that allow us to ensure that the community partners are doing things correctly. But there's always been the question about how well it would reflect what would have happened if the water sample had been tested by a core laboratory. And so this year, what we were able to do is essentially take the samples that were collected as part of the sampling program, and split them onto two different filters, which is a standard process for processing a water sample for qPCR analysis. And so we actually put 20 mils onto each of these two different filters that you can see on the slide here. And then one set of those filters went on to be tested using the traditional test that's used by Alberta Health Services to test for Enterococcus, which is built off of a US EPA method called Method 1611. And then the other filter was run by our group at the Alberta Lake Management Society lab that we established this year. And that ran a slightly modified version of Method 1611, where we did a few modifications to allow for it to be more amenable to be run by a community partner. In the end, the two different tests give you a slightly different readout. And the readout for the guideline test, Method 1611, is based off of a threshold exceedance value of 1,280 calibrator cell equivalents, CCEs, per 100 mils. I won't go into the details of how that's all done, but our test uses what we call a genome equivalent, or a GE value. And the biggest difference there is that we just can't have community partners culturing live bacteria as part of their qPCR program. So what we were really interested in understanding was how often are we able to predict a guideline exceedance of 1,280 CCE per 100 mils using our tests in a decentralized lab setting. And so what I'm going to present to you today is still a work in progress. We're still analyzing a bunch of this data because the program is just wrapped up. But I'm going to present to you data from 129 monitored samples, and 95 of those samples are included in this analysis to date. So we're going to talk about these 95 samples for the remainder of this part of the presentation. And our real question that I wanted to just address here was a very simple one, which is really just how often do our results using the genome equivalent value agree with the gold standard calibrator cell equivalent value with respect to the exceedance of that 1,280 guideline level. So in other words, when the gold standard test is over or under that guideline level, do we also predict an over or under on that guideline level? That's really what we're trying to address here, a very simple question, but it has to do with how we envision this program being useful in the future, which is really about sort of being a surveillance network that might help guide official testing. So never would it replace official testing, but might be able to guide official testing down the road. And so I'm going to just highlight in just some circles the results so far. So we analyzed these 95 samples in the gold standard test, and 38 of those samples exceeded the 1,280 calibrator cell equivalent value. If we took our test, we agree with that assessment 86% of the time. So 82 of our samples agreed with the results of the gold standard test. And when we didn't agree, we were almost always overestimating the gold standard test. So we had our 82 samples which agreed. The 12 of the ones that didn't agree were overestimating, and 2 were underestimating, which means that we ended up having 48 samples that exceeded the 1,280 CCE guideline as our estimate. So we're overestimating, but I think if we were to envision this as being a surveillance program that helps guide the gold standard testing programs, then I think we're overestimating in the right direction, because then we would be telling somebody that there might be a problem at a lake, where then when they do the gold standard test, there isn't a problem, rather than missing the problem completely, which would mean that there may not be a gold standard test even performed. And so we're quite happy with this, and we're still continuing to get more data on it, and we're going to hopefully learn more about this. But this was really a really incredible opportunity for us to understand more about the reliability of our community-based monitoring programs and how often we can predict an accurate result compared to a gold standard test, which is something that we often don't have because we could rarely coordinate the sampling and stuff like that would work out for this kind of a test to be done. So finally, I'm going to leave you with our vision for integrating education into something like this. And so I've shown you these two different stories that highlight the use of decentralized, democratized qPCR. And one of the ways that we think this could have long-term legs is to integrate it into an educational system where we can build on the biological parts of qPCR into training for students around DNA replication, for example, and we can then build the water monitoring component into a water literacy program. And we've had a lot of conversations about how this would look, and we see it as being a really interesting way to build not just the basics of water testing and the capacity to build water monitoring programs into school-based programs, but also teach students about how to understand science and ask scientific questions and understand the scientific method much more clearly. And so I'm going to return back to these models, and I think that the integration of schools into these models, the way that we envision it, really fits really strongly within Model C, where our vision would be that we would have teachers be trained by us or our partners within Inside Education, which you'll hear about. And those teachers would then be able to coordinate student projects that would eventually be able to take on research legs of their own. And so while the current model that we have looks something like on the right, where we maybe serve as this central hub that then has other hubs that connect to community partners, and it all kind of feeds in through a sort of a continuing condensation of the data, you could envision a situation where schools start to become their own hubs, and they can design their own projects based on the interests of students or teachers. And you could start to answer regionally specific questions related to water targets based on where the qPCR systems are actually placed. And so we are really excited about this opportunity because we think that it demonstrates a potential future for integrating qPCR-based water monitoring in a decentralized way, in a much more sustainable way than the potential for just community partners out in the wild to be doing it, which once we're pulling away from the project may not be something that's as sustainable as it is at a school. And so within that educational piece, we've made some really important partners. And you're going to hear from Catherine within Inside Education, which has been our most important partner in all of this. And we're really excited about continuing to move forward on this with them. But we've also had conversations within the University of Alberta and the Faculty of Education about ways that this might look. And we've also started partnering in Michigan related to the swimmer's ditch work I showed you with a college there that's going to start trying to run some of the qPCR samples that we've been doing here, run that over there so that we can start to have that become more of an autonomous program that is being run through a water literacy program and a water training program that they have there. And so with that, I'm going to wrap up and I'm going to let Catherine take over. And I'm happy to, I'll look at the Q&A section here and I'll try to catch up on all the questions. I see the little exclamation point there, and I'm happy to answer questions once Catherine's all finished. Thanks, everyone.
Thanks, Rick. It's really interesting to see the value of engaging with our communities and how they can contribute to research programs. A reminder to everybody to continue to submit your questions. They can be about the topic, about the research. We'll have some time for a good discussion at the end. Over to Catherine to continue the conversation on education.
Hi, everyone, and thanks, Patrick, for that excellent overview, and Vicki and Alberta Innovates for inviting us to share today. So my name's Catherine Wagner. I'm the program director at Inside Education. I've been with Inside Education since 2006, and a big part of my job and a favorite part of my job is the knowledge transfer. So usually this time of year, I'd be spending a lot of time going to attending forums, professional conferences, learning all about great work being done, and then figuring out how to share these stories with teachers and students across the province. And so that is kind of the goal for this presentation, is to encourage you to connect with schools and talk about why schools are such valuable community partners, and share a little bit about the work that we're doing with Patrick's lab and the water monitoring project to date. So a little bit about Inside Education. We are an environmental and natural resources education charity. We work exclusively with K-12 teachers and students across the province of Alberta. and really focusing on environment and natural resources. So water being one of the topics that we focus on, but we also do energy education, forests, agriculture, air quality, and kind of all the topics and issues and innovations related to those things, with the idea that students can play a key role in the environmental landscape here in the province of Alberta. How can they be great stewards for our natural resources, informed decision makers, and responsible citizens, and contribute to this landscape in the province? We were established back in 1985. And so have 35 years of experience working with schools across Alberta. And on an annual basis, we work with over 25,000 students in over 800 classrooms and hundreds of communities across the province. Our guiding principles are listed there on the slide, and really it's about showcasing the multiple stakeholders and perspectives present to enrich the curriculum with these experiences, these insights, so that students can really think critically about their role as environmental stewards in the province. And I should mention briefly that all of our programs are available at no cost to teachers and students. So we have classroom presentations, field programs, grant programs, resources, a whole bunch of stuff available for teachers and students to access on our website. So really, water literacy, and Patrick mentioned it, is kind of the overarching reason why we should all be excited to be here today, and why community-based monitoring is so important. So there's a definition of water literacy on the slide there, but you might have your own definition of water literacy. And to me, it's really about encouraging young people to be fluent in this language of water, like the root word literacy. So if students can understand the science, the topics, the issues, the innovations, and have meaningful conversations, then we can get to a place where we can think about water, the health of water itself, and the health of our environment, our economy, and our society. So researchers, we state our assumptions, and I teach students the scientific process to state assumptions. And I'm going to make the assumption that we all want this in some form or another, that water literacy is a goal for anybody on this call today. So how do we get there? Well, good water education will lead to water literate citizens. And so there's, the understanding, the science and the kind of textbook learning, but this messy arrow that leads to the understanding, to the action, to doing something about it, to contributing to this landscape, that's where the work I do takes place, that zigzaggy arrow between water education and water literacy, the zigzaggy arrow between understanding and action. And that action can look different. It could look like contributing to a community-based monitoring program, could be a student pursuing a career in the sciences, it could be a stewardship project, or even just going home and telling their family, friends about what they're learning in this water space. And there's some key things that Inside Education incorporates into all of our programming. So water education, or insert other topic area, but really it's about having locally relevant, meaningful conversations, thinking about their interaction, and importantly for Inside Education, how it connects to the Alberta Program of Studies or the curriculum. And there are so many ways. Patrick mentioned molecular biology. We can go to the water cycle, watersheds, but we can also go on the social studies side with water management and policies, and the list is endless. There are multiple connections to water education throughout the curriculum. And we're really lucky to be working in this space for that reason, as well as that water is really tangible. It's easy for students to connect with, and they can see it, they can feel it, they can interact with it. So we have a really good launch point to get to this water literate society that I'm assuming we are all striving for. Okay, so how do we end up with the community-based monitoring and the qPCR technologies that Patrick's talking about? So I'll tell you a little bit about how this partnership came to be. Inside Education has been very fortunate to work with Alberta Innovates for many, many years. And one of the programs we offer are teacher professional development programs. And back in 2017, I was hosting a water program for teachers. And I reached out to Vicki and said, oh, we're looking for a great tour somewhere where we can showcase a really cool story about water innovation and meet with some experts. And Vicki pointed us in the direction of Patrick's lab. And I'm not sure Patrick quite knew what he was getting into when he agreed to let us a tour at the U of A. But we showed up on his doorstep with 25 super keen and eager teachers, taking a bunch of pictures, asking a bunch of questions, and really getting excited about the work that was happening in the lab at the U of A. And one of the things that he mentioned while he was there is this community-based monitoring program and access to data collection. And that's sort of where the light bulbs went off, that Inside Education, through our current water education programs, that we were going into classrooms already and had this vast network of schools, that schools might be a really great place to implement some of the community-based monitoring and that is happening. And so with that, sort of back in 2017, this partnership grew from there. And citizen science or community-based monitoring is really where the magic happens in a partnership like the one between Inside Education, Alberta Innovates, and the U of A. And Patrick talked a lot about the different models and what this means. But to me, the exciting part of it is that citizen science or community-based monitoring can be a form of simultaneous learning as well as knowledge generation. So there's tons of mutual benefit and value. And a big take-home message here is that students and schools and teachers want to be engaged. This area is growing, and there's so many opportunities to get students excited about this. As an example, one of the things we did early on in our partnership was Patrick came to a youth summit program we were offering, and we asked the school teams coming from across Alberta to bring a water sample so we could, practice with the qPCR and the extraction of DNA. And again, these students were coming from all over. And so they brought, you can see on the bottom picture there, water samples that they collected in March. So not only did we get the bottle of water, we got the stories of them cracking through the ice, trying to get access to their stormwater ponds or local water bodies so that they could contribute, because they saw a lot of value and they were very excited about, adding real data and meaning to a project like Patrick's. And I'll add in here as well that it really doesn't matter what age of students we're talking about. These were high school students and the molecular biology DNA side of things. But even my kids, through our own recreation, have gotten swimmer's itch. And so they would be excited to know if that was something present in their water bodies. So a lot of meaning and purpose can come from community-based monitoring with schools. From the kind of external looking inside, why work with schools? it can add a lot of value to your research. So schools are everywhere. The map on the right shows Inside Education's program reach in 2019. So we work directly with each one of those schools, and a lot of opportunity to increase the geographic reach of your research questions or parameters. And there are over 700,000 students in Alberta that have the potential to be excited about what you're doing and are excited to contribute to what you're doing. The trouble is that they often don't know what you're doing. And so that's a big role that Inside Education plays, is connecting the industry, the sector's research to local schools. There's also the benefit of curriculum. So Alberta has a program of studies of which teachers follow. There are tons of opportunities to connect to water, as well as other topics. And the neat part about it is that it cycles annually. So teachers will be teaching that unit in curriculum over and over and over again. So you can really maximize the education value there, as well as having data on an ongoing basis if you're using for data collection. Or, as Patrick said, to have the schools think of a question and really add some meaningful education pieces to the current Alberta program of studies and the lessons that teachers are doing. Another benefit is that schools are community hubs. So they have vast networks that could be as easy as a student going home and telling a parent what they learned that day, something that they did, or hosting a showcase, or maybe bringing the QPCR out on a school project or day. And you never know where those connections and where those networks might lead. Maybe new research questions, maybe new partnerships, maybe additional funds for what you're working on. Patrick kind of talked about this. It allows you to scale a project both geographically, as well as maybe different research questions with minimal cost, and the water literacy side of it to increase your impact by sharing your work and telling your story. So the overarching goal of the collaboration between the U of A and Inside Education is to help schools be community hubs for water monitoring, to get those CHI QPCR machines into select schools so that they can start to create their own research questions, as well as contribute to the work being done at the U of A. And we are building towards this through our programming. And currently we have 3 different approaches that we're using to connect with schools. So teacher training, student sessions, and school projects. And I have one slide on each of those just to tell you a little bit more about how we're doing that. So the first area would be teacher professional development. And I would encourage you, this is a great place to start. If you're interested in working with schools, you can either come and let Inside Education knock on your door and bring 25 teachers into your lab asking a bunch of questions and taking pictures. But one of the other things we're doing is, this is a photo of my colleague Andre teaching these teachers how to use the machine and the value of doing that. And through training programs like this, we can use this train the trainer to extend the reach and the possibilities for schools. And teachers are really excited to start incorporating some of this, not only to enhance the curriculum and for the water literacy education goals, but also so that they can enhance what they know and access to experiences and experts that really bring that curriculum to life for their students. And one of the big roles that Inside Education plays is helping teachers understand how to incorporate that, something like QPCR and DNA analysis into the curriculum. And so we spend a lot of time workshopping that on single or multi-day events where we're working directly with teachers. And Patrick mentioned briefly, there is a researcher working on training videos and materials and curriculum pieces that will augment this teacher professional development aspect. Another thing that we're doing is working directly with students. So Inside Education has 3 CHIs, or 3 qPCR machines, that we bring to classrooms across Alberta. And it's a really cool program. Teachers get really excited about it because we ask the schools to collect a sample of water from a local stormwater pond or a water body. And then we bring it into the class. We talk to them about what they think might be living in there. Are there invasive species, species at risk? What do they know about that water body? We run it through the CHI. It takes about an hour to run. And so in that in between time, we can do a lot of water education pieces with them and have different activities and things we do with the students. And in the end, they get to see these real-time results of what's living in there. So it teaches the scientific process and gets them really engaged in their own backyard, in local water quality topics and issues. And another neat part is because they book it in advance, we have the ability to work with Patrick and say, oh, hey, we're doing a program in this region of the province. Can you send specific primers so we can look for specific research targets or specific things that the students would want to look for, like, for instance, goldfish in St. Albert wetlands and ponds. So a lot of value that way. And then this is a big area and connected to our youth summits. One of the things we do is working with school environment clubs or school teams to develop projects and programs. And we're hopeful that we can then have them have their own CHI or qPCR that they can start asking their own research questions, as Patrick mentioned. And Inside Education works with them in the project development and planning and how we're going to, what that might look like in their particular school situation and location. What kind of questions they might want to ask: invasive species, species at risk, are there biological hazards? So it also provides the opportunity for a lot of peer-to-peer learning. So then this student group that's analyzing the water on an ongoing basis can then share it with the teachers and students in their school community, which then increases the impact of that work, as well as extending the education and water literacy value of the work that we're doing. So those are sort of the three ways in which we integrate qPCR and community-based monitoring into schools across Alberta. And because I'm a teacher by training, I get to assign homework at the end. So homework for all of you listening in today would be to consider the mutual benefits of working with a school in your community, and then reaching out to them. And if you're not quite sure how to do that, you can connect with Inside Education, but it can be as simple as, if you have a child of your own and you want to go just have a conversation with that teacher, I'm sure they'd be more than excited to hear about what you're doing and figure out how to connect. And then you never know where those conversations and connections might lead you. So you'll end up with new champions of your work and partnerships. So with that, I think that kind of wraps up the value of working with schools. I hope you realize how important it is to connect with young people, the water literacy connections. And I hope you're inspired to go out and make those connections of your own. Thank you. Happy to answer any questions as well.
Thank you, Catherine. That was great. Great overview of the value of connecting schools. I know personally, I've had the privilege of participant inside education events and meeting with teachers, and the enthusiasm for this is really amazing. And I think a largely untapped group that we can connect with as a research community, to really engage and realize that there are future researchers, there are future decision makers, and the earlier we connect with them and get them excited about this, the impact that we can have on our water, and as you mentioned, other topics in the province too. Questions, and you're welcome to continue to submit questions as we go through this. I'll continue to scroll through and bring them up for the speakers to answer. I'm going to start with one here. So this, yeah, but it's about how do you start a community-based monitoring program? How do you recruit volunteers, participants? Are there guidelines or a manual to help to start a study? Do you require minimum education or knowledge participation? So a bit of a multi-part question there. I'll start with you, Patrick. I'll get you to chime in on it as well afterwards.
Yeah, sure. It's a great question. I probably could give a whole presentation on how we've managed to get partnerships going so far. You know, often it kind of originates kind of organically. Presentations like this have actually been a really great way that we've ended up getting partnerships off the ground. Either, somebody from my group or myself will be giving a presentation somewhere and somebody will come up and say that, sounds like something we'd like to be part of. Or sometimes it's the other way around, where we hear a presentation from somebody doing something really interesting, and we see a way that we might be able to integrate DNA-based testing into it. That being said, we also have really tried to leverage some of the partnerships that other partners that we have. So Alberta Lake Management Society, Alberta Environment in Parks, and groups like that often are really great resources as well. And as we start getting them more integrated into the DNA-based monitoring programming, then those also become venues for making some more partnerships. It's a little bit tricky sometimes because we often find that everybody would like to have a qPCR machine, but it becomes slightly cost prohibitive if we envision trying to have one at every single lake in Alberta. So sometimes it's about trying to find a centralized venue to place it so that it can serve sort of a bit more regionally. And that's where we've often thought that schools might be a really great way to sort of house the infrastructure that would be required for an area to be able to access qPCR. In terms of the other parts of the question, though, I think Catherine mentioned this. We actually just recruited a master's student who her entire thesis is about training people to do qPCR, and so we've created a whole bunch of videos that span between the theoretical underpinnings of qPCR and just water monitoring in general, all the way to very hands-on practical training videos about, you do this step first, you do this step next, and keep going. And so we do have sort of written and video-based manuals for people to follow. And part of her thesis is actually evaluating how well people do after some of that online training versus hands-on training and stuff like that. So we can understand a bit more about what are the best ways to take somebody who has never done molecular biology before, which is most people, and get them to a point where they're confident and capable of doing things relatively accurately. And you don't need a minimal education or anything like that. We try our best to make all the resources very accessible so that people can access them and understand what's going on at each different step, and understand what they're seeing when a qPCR machine is finished running. And so far, a lot of that has worked reasonably well. So we've had quite a bit of success getting people to the point where they're relatively competent at running qPCR. And like most things, it's certainly, you get better at it with experience. And so the first couple times you run it, maybe you're not as good, but it doesn't take long to get better at it. So yeah, definitely it can be tricky to find all those partnerships and make it so they're the most sort of most effective. But that's all stuff that we're working on now as we try to progress into the next couple of years of this project, and really start to get more of this out into the communities in Alberta. So, yeah.
Thanks, Patrick. Catherine, you did touch on it a little bit in your homework, but from a community member perspective, how would you encourage researchers to reach out and engage?
Yeah, so when I work with teachers through our teacher professional development, I tell them the same thing: find researchers, find cool projects that you can connect with. So on the flip side, it would be doing the exact same, having researchers or folks that want to get into community-based monitoring reach out to schools that make sense for their project. I think there's a lot of willingness and enthusiasm and excitement, and they'd be more than happy to connect. I think we're often just scared to pick up the phone on either end. So really important to do that. And I will reiterate what Patrick said, the training is critical to that. Until, often we might not have the understanding of, the back end, all the research that's happening. But if we can implement a really good training program, either through a professional development or hands-on workshop, that will go a long way towards the success of the project.
Thank you. This next one, we'll go more to this a little bit in your presentation, but can you speak to the reliability of data that's collected through these types of programs?
Yeah, sure. I can talk a little bit about reliability.
Or others' immunity?
Oh, sorry. You broke up there a little bit, Vicky. I didn't hear the last part.
Reliability of the data and how we can make sure that it's up to by regulators and others in the science community.
Yeah, that is a really good question, and I think it's one that is more or less important, I find, depending on the targets you're looking for. So, one of the reasons I shared the story of the enteric bacteria stuff that we did this year is because that's kind of at the upper echelon of scrutiny for data reliability. Typically those types of samples are run in a very controlled lab in the Provincial Lab for Public Health, and there's a really defined sort of pathway that those samples are processed in a certain amount of time. If something happens, then, and that leads to a beach advisory, then there's a certain protocol that has to be followed to follow up on that. And so those are pretty rigid and rigorous protocols that are followed. And I think that it is important to be kind of realistic about where a community program can fit into something like that compared to a more investigative kind of research project that might be about understanding, what are some of the hazards associated with storm ponds in Edmonton or something like that, where there's a bit more flexibility, and you might find something really interesting, but the stakes aren't quite as high as a beach advisory, for example. And so, like I said when I presented that part, I think, our vision has always been that something focused on, for example, enteric bacteria might fit in as a bit of a surveillance network. It wouldn't necessarily ever be thought of as replacing the gold standard test programs, but it might help direct those programs and maybe make a more efficient use of the resources that they have. Because as you can see, even in the gold standard test, only about a quarter of the samples test back was positive. And so a lot of those other sites that are tested negative, if we were able to help target the sampling that was happening for the sites based on, a network that we had in place of community-based partners, that might help just target the gold standard program much more effectively. So in terms of reliability, it really comes down to making sure that, like Catherine said, that the training is in place, and that the person who is doing the qPCR on the community side is somebody that's trustworthy. And the way we get to that point is often about doing a bunch of essentially blind samples with them, where we use samples that we know the answers for, and we see if they get those answers, and we make sure that they can do things like extract DNA and filter the samples and amplify the targets well, and get the answers that they should be getting using controls. And then we start getting to the point where they're running investigative samples and stuff like that. So it takes a little bit of time to get the community partners up to speed, but once they are up to speed, we've often found that that they're usually quite reliable after that. So we have data on a couple of different partners now that have been partners with us over a couple of years, and that usually is the case where, you know, there's the first couple weeks or months maybe where there's a bit of variability in their samples, but then after time they become a much more reliable qPCR partner. So yeah.
I think that leads into the next question here is training, and how do you ensure the longevity of programs like this? So your research may only be a couple of years, or you want to make sure that those community members stay engaged. So how can you speak to that?
Yeah, and I think this touches on something that you know, certainly in Catherine's realm of understanding better than mine, but you know, we've kind of straddled this world of having people in the community running qPCR, and then we also have the school partnerships. And one of the limitations is that, you know, for the next few years, we're certainly engaged and able to help coordinate and get things going. But the goal would be that eventually this becomes something that's self-sustaining, and then the partners that we've trained are able to continue to run qPCR based on a financial model that works. You know, one of the things that we advocate for is that qPCR isn't particularly expensive to do when you compare it to other water monitoring techniques, but it is expensive to contract others to do, because there's a lot of costs associated, especially with having expert people running qPCR. So financially, it's not prohibitive necessarily, but there needs to be some guidance, and that needs to be something that happens over the early parts of the partnership to make sure that everybody is on the same page about what the expectations are once, you know, for example, once we start becoming less involved in designing the project and more about just kind of making sure that everything is working properly. And our goal is always to kind of start pulling away once we're confident that the partners are capable to do qPCR. We start becoming more about trying to coordinate larger research questions and making sure that the quality is there continuously by just running a few samples from community partners kind of randomly as a sort of a quality assurance program. But I really think that in terms of longevity, the school partnerships are really appealing, because the school partnerships are where you can have not just the infrastructure housed and the potential for those programs to run as part of school-based programs, but they're also where you can have the knowledge housed with teachers who are probably a lot more persistent within the community than some of the community volunteers who might come and go during the year or something like that. So we really see the teachers as an important resource and point of training, where there can be a lot of knowledge sort of stored in that resource of teachers that can then pass on to others who are using the infrastructure within the different regions. So yeah.
Well, that's a good segue to Catherine. Maybe you could comment a little as well. I think there may be some concerns from some researchers about the longevity with student partners, because they're only in the system for so long. So the concern about that versus perhaps the value of the, as Patrick mentioned.
Yeah, I think that connects to the curriculum piece, the program of studies, that isn't going anywhere. Schools are consistent. They, you know, that cycles annually what they're teaching. And if we can use training programs to encourage teachers to incorporate that into their lessons, that will become an integrated part of what they're doing. And I think I mentioned that the school is the community hub. You know, it's a connection to parents and kind of the broader network that surrounds a school community. So it's there's a lot of longevity that comes with schools for that reason. And then school environment clubs are great, although
Absolutely.
the students themselves might turn over, the environment club or school team would stay there.
Absolutely. Then last question here. I don't see too many more coming in. Patrick, is more a comment than a question, but just the value of potentially reaching out to other groups like the WPACs They have education coordinators, or perhaps you could mention some other groups that you see value in connecting with that maybe you haven't to date, or perhaps other researchers might want to consider.
Mm-hmm. Yeah, it's a great comment. I mean, this year was a little bit kind of strange for us because our initial goal was really to start reaching out to a bunch of WPAC groups and other lake associations and things like that early this spring to try to start placing qPCR machines regionally that made sense. So we realized that we can't provide every lake with a qPCR machine, for example, but within sort of regional priority areas, we can certainly do our best to try to get some of these things out there. But then COVID came around, and the company that made the chai stopped working for a little while, so we couldn't get those. And so we kind of had to adjust a bit of our expectations for this year. And so that's something that this fall and winter, we're definitely, now that we know we can start getting the infrastructure up and running, the goal is to start reaching out to different groups and gauging interest and figuring out where we can place things based on some of the priorities of our project, and also the priorities of those groups in terms of what they want to look for and answer questions they want to answer. And so we're not restricted in any way in terms of who we might want to try to partner with with that respect. I think that we would like to have conversations with anyone, and then it might just come down to what makes most sense to be most effective with the resources that we have to try to create a comprehensive program that's testing for sort of at least geographically relevant targets in each of the different regions that we can place a machine. And so those are sort of our sort of things on the priority list for this coming fall and winter, is to try to get all that stuff set up so that we can have the training occur before sort of the ice comes off the lakes, and have people prepared to do some sampling and running qPCR early in the spring and summer of 2021. So, yeah.
I know there's a lot of value in science of what you're doing as well. We've seen the growth of eDNA in our water system. Your team can be with whirling disease is a great example that you guys added on to your work. So I think from a science perspective, away from the great to see what your team is doing, and thank you so much for sharing. And Catherine, your experience, and like I said before, I think it's a really untapped potential to engage with the water literacy and the water passion. Choppy here, so hopefully you can hear me close it out. But thank you everybody for attending today's session. Just to spark some new ideas, you have some connections that you want to make. The session is recorded. It will be posted on our website in the next week. We will be sending out shortly after the session management. What have we learned and where are we going? October 22nd. The full schedule. Click the button in the top right corner of your screen. We're connecting with you again, and all the best. Thanks, everybody.
Thanks, everyone.
Thank you.